Welcome to my YouTube channel. So here today we are going to be looking at lower fungi. So fungi is grouped into what we call lower fungi and higher fungi. So here we are going to be looking at the first lower fungi, which is Zygomycete. In another video we'll look at Mycete. So now what is Zygomycete? So under Zygomycete we can have an example such as Rhizopus. So we can have an example such as Rhizopus. So this is able to produce Zygospores. So they are able to produce Zygospores. So we'll understand further what a Zygospore is, but just a brief idea of what a Zygospore is. So a Zygospore is a thick word resistant sexual spore that is formed by the fusion of two compatible gametes or hyphae. So we have a structure like this. This is hyphae. So when two hyphae come together, when they fuse, they produce a spore which we are going to call a Zygospore, like that. So they will be able to form a Zygospore. So Zygomycete is able to produce a Zygospore, just as the name entails. Zygomycete, you can just think of it as being able to produce a spore. So by definition, that's for those that are taking note. So this is what we are simply saying. So a Zygospore is a thick word resistant sexual spore formed by the fusion of two compatible gametes, or rather hyphae. So the other characteristic of Zygospore is that the hyphae is usually haploid, meaning to say it contains half the number of chromosomes. So now they are also unsegmented. They do not have segments, that's the meaning. So they are unsegmented, like that. So that is what we mean. They are not having segments like that. Now when we look at the other things, so by definition, hyphae is a thread-like structure. So this is what we are saying. So hyphae is a thread-like structure that serves as a basic building block of any fungi. So that is basically what hyphae is. So now when a group of hyphae come together, they will be able to form what we are going to call mycelium. So when a group of hyphae come together, they will be able to form. So these are thread-like structures which we are going to call hyphae. When they come together, they will be able to form what we call mycelium. So mycelium is what is formed. Since they are unsegmented, you might have heard of the term that they call coenocytic. So we have the term coenocytic. What does it mean? So when we say coenocytic, we are going to say that they have a lot of nuclei but lack a cross wall. So this is what we mean when we say they are unsegmented and they are coenocytic. They have a lot of hyphae. Sorry, not they have a lot of hyphae. They have a lot of nuclei but lack a cross wall, like that. So this is what we refer to. As you can see, all these are nuclei. That is what we mean when we say they are unsegmented. But if those that are segmented, they look like this. So if we are looking at septate hyphae, meaning to say they have segments like this, like that. So like that, you will find that this is what we mean when we talk about like this. So you can have the nuclei like this, or like this. So this is what we refer to as, this one is septate. Septates, meaning to say they have separations like that. But for Zygomycete, it is unsegmented and it is coenocytic. Coenocytic means to say they have a lot of nuclei without a cross wall. As you can see here, there is a cross wall. This is not Zygomycete. This one is Zygomycete, or rather, this is also unsegmented hyphae. So this is unsegmented hyphae, whilst this other one is segmented because it has got a cross wall like that. Now, with those characteristics discussed, we are going to now look at sexual reproduction. So under sexual reproduction, the first thing that takes place, we have all these spores are on Rhizopus are haploid, meaning to say they contain half the number of chromosomes. So then when we have this is what we are going to be calling release. So they will be able to release the spores like that. So these are our spores. Then the spores will be able to grow and become hyphae. So this is hyphae. So other books will tell you mycelium, but remember, mycelium is just hyphae coming together, a lot of hyphae to form a structure called mycelium. So the spores here, we are going to say the spores germinate, or rather the process is germination. So germination, the spores become spores to hyphae is what we call germination. When that happens, through the process of mitosis, there is the formation of sporangia, or rather sporangium. So this part is what we call sporangium, the one that is able to produce spores like that, and the cycle continues. So asexual reproduction, which means without any sexual activity, is quite easy. Now with this understanding, let's now look at sexual reproduction. So under sexual reproduction, it's quite a bit different. So now we have the same thing. We have the first stage, which is our release. So we are beginning from here. So we have the sporangium release the spores. So sporangium are the ones that bear. So this structure is sporangium. So the sporangium will be able to, more like I would say, produce spores. Then the spores are released. When the spores are released, they begin to germinate, just like sexual reproduction. Now when they germinate, what then happens is that the hyphae, so they will germinate into hyphae. So we have the hyphae like that. Then this hyphae will be able to specialize and become gametangia, or rather the male and the female sex cells. So it will be able to form gametangia, which is the male and the female. So for Zygomycete, we just use the plus or the minus. So that's that for gametangia. You use the positive and the negative. We don't use anything else. So this specialization is what we are going to call differentiation. So during differentiation, they were the same hyphae, but they have specialized to become male and female. So now when these male and female fuse together, they will be able to undergo plasmogamy. So plasmogamy, just as the word entails, plasma has to do with a liquid. So plasmogamy is what is formed when the two fuse their cytoplasm. So plasmogamy is the fusion of the cytoplasm. So this leads to the production of an heterocyte an heterocytic cell. The heterocytic cell has got the nucleus. So this one, remember, this hyphae has a nucleus and this hyphae has a nucleus, like that. So they have a nucleus from both male and female. So this type of cell is what we call an heterocytic cell. So this cell is, so it is after this fusion. So this is plasmogamy. Now I've just drawn this one to allow you to understand what goes on. So this is, the cell is called heterocytic cell, like that. So this heterocytic cell will now be able to undergo the process of karyogamy. So in case you are not seeing it properly, that word is karyogamy. So this karyogamy is the fusion of the nucleus. So the fusion of the nucleus is what we call karyogamy. So after karyogamy, there is the production of what we call the zygote. Then after that, the zygote will be able to undergo the process of meiosis. So if you cannot see this word, is meiosis. So meiosis is where the number of chromosomes are more like reduced to half. So after meiosis, there will now be the formation of sporangium again, and the cycle continues. Now remember, one of the effects of meiosis is that there is the production of genetically different spores. So since we are dealing with spores, there will be the production of genetically different spores. So that is the essence of sexual reproduction, and that wraps up sexual reproduction. So just a recap. So we have sporangium. It is able to produce spores. Spores are released, number one. So these spores will begin to germinate, or rather to grow, to produce hyphae. Now this hyphae is the same. After that, the hyphae will undergo the process of differentiation to produce the male, which is the negative, and the female, which is the positive. So under Zygomycete, we just refer to them as positive and negative. Then from there will be the process of plasmogamy, in which these two fuse. When they fuse, they form what we call an heterocytic cell. This heterocytic cell contains both the male and the female nucleus, like that. Then after that, when these are like this, so plasmogamy is simply the fusion of the cytoplasm, so which leads to the production of an heterocytic cell. This heterocytic cell will now be able to undergo the process of karyogamy. Karyogamy is the fusion of the nucleus. After that, then we are going to have our meiosis, in which there will be the reduction in the number of chromosomes, and thereby producing half the number of, or rather genetically different spores. Then we are going to produce the sporangium, and the cycle continues like that. Okay, so others might use mycelium. Mycelium, remember, is just the fusion of many hyphae, like that. Then we are going to have, from mycelium, we are going to have plasmogamy. That is after, if we have two different mycelium, they fuse together, the positive and the negative. Then we are going to have plasmogamy. Here it's saying that haploid cells from two different mycelia fuse to form an heterocytic cell with two or more nucleus. Remember, these nuclei are more like separated. After that, then there will be karyogamy. Karyogamy is where the nucleus fuse to form a diploid zygote. So we have just one nucleus, they have fused together like that. Then we are going to have meiosis, in which there will be, from zygotes, there will be the haploid spores are formed, which are genetically different, like that. Then when the haploids form, there will be germination to form mycelium. Remember, mycelium, a lot of hyphae, like that. the cycle continues. Then this is asexual reproduction. For it from this stage, when they are not specialized, they will be just able to go mitosis. Mitosis will produce the spores. Spores will germinate. The cycle continues like that. So that's basically it for this video, and stay blessed. So that is that under this video. See you in the next tutorial.